One thing I should point out is that our textbook and a number of different institutions and professionals will call dehydration synthesis, they'll call it a condensation reaction. So the two names are basically interchangeable. You'll see some places will refer to it as dehydration synthesis, which is the name I prefer, and then some people call it condensation reaction. Now, I think dehydration synthesis is the better term because it really describes what's happening very well. It's very descriptive. D, removal, hydra or hydration is water, removal of water, which is exactly what happens. Synthesis is the creation of a larger molecule, whereas condensation reaction refers to the formation of water, like when water condenses on like your car window or something like that, or in the mirror in the bathroom. So yeah, that also refers to the formation of water, but reaction doesn't really tell you anything about the creation of this larger molecule. So nevertheless, this is a common thing that, or a common name that's used. Like I said, the Campbell textbook uses this. I tend to call it dehydration synthesis. You need to know that the two are the same thing, because you might see either one on an AP test question. All right, this is just like a more beautiful diagram of the scribbles that I was making. Some graphic artist created this, and it just shows you that in dehydration synthesis, smaller molecules can get linked together. Water is going to get pulled out, so we got our HO, we got our hydrogen, that gives us 2 hydrogen and an oxygen, and it's going to end up forming water. And then we get a larger polymer that ends up forming from this. Okay, and then many dehydration synthesis reactions are going to form these long polymers. It has to happen many times for that to happen. Hydrolysis then is the opposite, and that's going to get split here by the insertion of water. So water is going to come in, it's going to split this up. And by the way, water by itself does not do this. Water does not have that power. Enzymes will help. There are enzymes involved in this that make this happen. And the enzymes we kind of leave out of these diagrams and these explanations because we want to simplify it right now. We want to see just what's happening with the water. And now the hydrogen goes over here then, and the hydroxyl goes on this side. So keep in mind that the larger molecule, the polymer, gets split, and the water gets split also in hydrolysis. And hydrolysis is a good term. Hydro, water, lysis, splitting or breaking. All right, it's time to talk about carbohydrates. So that's the first type of biological molecule that we're going to discuss. And carbohydrates sometimes also are known as sugars and starches. And there's other carbohydrates also that we're going to mention, like cellulose and things like that. We'll get into that. All right, so we know that carbohydrates are frequently long chains. They're frequently polymers that are made out of individual monomers that we call monosaccharides. I don't know if I can squeeze that in up here. Let's try it. Might have to split it up here. Monosaccharides. So these are a bunch of different monosaccharides. They distinguish between aldoses and ketoses. I'm not too worried about that. That refers to whether the base molecule is an aldehyde or a ketone. So we're talking about aldehydes having the double bond O at the end of the molecule here at the last carbon, or the first carbon, I guess you could call it. And then in a ketone, the double bond O is, or the carbonyl, I guess you could call it, is in the middle of the molecule. It's not on the end. And that just gives the sugars different properties and makes them slightly different. It's not that big a deal for us. Ribose is one you might recognize. It's a 5-carbon sugar that's in RNA. Deoxyribose is the one in DNA. That's real similar to this, just one of the O's is missing. That would be a pentose because it's 5-carbon. This tiny one here, glyceraldehyde, we'll run into that later as a triose. That's kind of a weird one, kind of a bizarre kind of sugar, but still considered a sugar just because of the biochemical definition. But the most common ones we're familiar with are hexoses that have 6 carbons. And that's stuff like glucose, galactose, stuff you've heard about before. Fructose, you've heard of, like, high fructose corn syrup. That's in our food. Ribulose will actually run into later when we talk about photosynthesis, and dihydroxyacetone will actually run into also when we talk about glycolysis quite a bit later. So what I want you to notice here is all these monosaccharides listed here are linear. They're drawn out in the linear form, which is, can I draw on this? No, I can't. Okay. They're drawn out in the linear form where we're used to seeing monosaccharides as rings. So what's the difference? Well, the linear form is what they're like as a crystal when they're a solid, not dissolved in water. The ring form is what they form when they're in water. So that's really more like the normal state of the sugar. Anytime in a biological system, we're talking about, in a body or, in an organism where there's also water. So they'd be dissolved in water and they would inevitably be in ring form. The only time they're in linear form is when they're like, sugar crystals in your sugar bowl, at Starbucks or whatever. Anyway. So it just shows how the linear structure can go to the ring structure, and this is really the one that we know and love. That's what we're familiar with for glucose. And this happens to be alpha glucose. So this happens to be alpha glucose, which is the one that we eat. That's the one that's in our food that we can digest. And it has the hydroxyls starting with carbon 4 on the left here, going down, then going up, then going down. This one went down, up, down, and then this one goes down. All right? So the reason these are in blue, and it just shows you where it was connected, like where it came from. It came from this one over here. That doesn't really matter. I'm not going to hold you responsible for knowing this reaction. I really don't care about the linear form of them at all. I just want you to realize that does exist, but it's not super important to our purposes. The ring form is the one we care about. All right, keep in mind that this H2 up here really refers to hydrogens being stuck out here. Okay, so those hydrogens are like right here. It's just an abbreviation, that's all. And also notice that the carbons are numbered, just like we numbered the alkanes. So we got carbon 1, 2, 3, 4, 5, 6, because it's a hexose that has 6 carbons. And the 6th carbon sticks out like an antenna. It's like this extra piece sticking out on top, and all the other ones are in a ring. And one of the corners of the ring is oxygen. So anyway, so let's practice drawing that. You should draw it also. I'm going to draw it, and then I'd like you to memorize that structure. It's pretty easy. You might have done that freshman year. If you're in my class, you most certainly did. So I'm just going to write a hexagon. I'm basically going to draw out a hexagon in my mind, and then I put it on paper or on the screen. And I put the top right corner of the hexagon, I make that an oxygen, and all the rest of them I'm going to make a carbon. So that's kind of easy. Just make a hexagon. It's easy. There it goes. And then I'm missing 1 carbon, so I stick that up here. That's carbon 6. So now I got to have the backbone. The backbone is pretty much done. So then I need to put my hydroxyls in. Here's what gets a little bit tricky. I just have to remember the down, up, down, down positioning, and then I stick them on here. So I just draw those in, down, up, down, down, and then we're good to go on that. I suppose if you start with carbon 1, it would be down, down, up, down, so that works too. Whatever you want to do for yourself to remember it. This hydroxyl you could put anywhere, so you could have put this on top or on the left, it doesn't matter. And then these get filled in with hydrogens. I just put it here because it looks nice. This is hydrogen, a lot of people forget, but each carbon has to be happy. It has to have 1, 2, 3, 4 bonds to it. This one has 4, we're all good to go. This one still needs 1 here, so we fill that in with hydrogens. So what you may notice after you're all done drawing it, if you count the carbons, the hydrogens, and the oxygens, here's what you get. You get 6 carbons, you get 12 hydrogens if you count them, and you get 6 oxygens. So if you reduce this down to the empirical formula, which is, sophomores, you don't know what that is yet, but the simplest, most reduced version of this, the simplified version would be just 1 carbon and then 2 hydrogens and then 1 oxygen. Okay, so if I divide all the numbers by 6, essentially is what I'm getting, I get 1, 2, 1. So what you may notice is that for each carbon, you then have a H2O. For each carbon, you have a water stuck to each carbon, essentially, is what it is. So notice what these things are called. They're called carbohydrates. Well, what does hydrate mean? Hydrate means water. Okay, so carbon water basically is what this means. How nice. Sugar is actually carbon water. That doesn't sound as appealing. Anyway, that's what it is. This proportionality, this 1 to 2 to 1, or one water for each one carbon, only works in monosaccharides. So this only applies to monosaccharides. It does not apply to disaccharides or polysaccharides. And the reason why is because when they link together, a water gets removed, which means if I'm going to link two glucoses together, let's take a look at that. We're going to have C12, and then you might say, well, it's going to be H24. But it actually isn't H24, because to link the two together, we had to take H2O out. So I'm removing two hydrogens. That leaves H22. And the oxygens doubled, but then I pulled one out. So it's really only oxygen O11. Okay, so C12H22O11, I'm not asking you to memorize that, but so that would be, maltose with two glucoses stuck together. It no longer fits that proportion of one carbon to two waters. It doesn't work anymore. You have actually less because we lost that one water with dehydration synthesis. So anyway, the whole like one to one thing with carbon and water only works for monosaccharides. And some people make a big deal about that. I don't really think it's that important, but I thought I'd throw it out there because sometimes I see test questions that really dwell on this, like it's some kind of big deal. I don't know. Some teachers really emphasize it a lot, so I thought I'd mention it. No big deal. All right, well, what we're going to have to do is link two of them together now and actually draw it out. So what I'd like you to do is on paper, or if you have a way to draw with a tablet or something, a touch screen or whatever, is draw two glucoses. Okay. Draw two of them and then link them together. And eventually I'd like you to be able to do that by memory, so I would like you to do it without having to look up the structure of glucose. But keep in mind, here we got that OH, here we got that hydrogen, and that's where the linkage is going to happen. That's where the dehydration synthesis is going to happen. Okay, one other thing I should probably point out is why am I sometimes writing these as O, and then sometimes I'm writing them as HO, and I know people like to laugh when I say HO, but okay, it's just a garden tool, okay, to rake my lawn or whatever. Anyway, or to till the soil, I guess. Why do I write it like that? And this goes back to the whole valence spiel. We said carbon has a valence of 4, like, each carbon has to have 4 bonds, like this one does, and all of them do. Oxygen can only have 2 because it has a valence of 2, so it can only have 2 things connected. So this really would be, would really look like this. It's COH. I'm just too lazy to draw it like that. It doesn't look as nice. So this OH right here is actually O with a covalent bond H. Well, this one over here is really this. It's HOC. Okay, so the oxygen has 2 things connected to it. There's 2 bonds. The hydrogen has 1 because it only has a valence of 1. It can only have 1 linkage. And then the carbon has its 4, and it's happy. If you did it the other way around, if you wrote this as OH, here's what would happen. You'd have the oxygen here with only 1 covalent bond. Then you'd have a hydrogen, which then would be forced to link to a carbon. And we're using hydrogen as a connector. Well, that's actually not possible. Hydrogen cannot be a connector. It can only be on the outside. It can never connect 2 other atoms. So this is wrong. It can't be like that. So the proper way to write this over here would be HOC, because this carbon is connected to this oxygen, and this oxygen is connected to that hydrogen. All right, over here, this is fine because it's once again the oxygen in the middle, the hydrogen on the end. No problem.